After replication, now we are heading towards the transcription process. First, we discuss. First, we made the base of the transcription process, splicing process, and then we will discuss them in just a simple diagram. So, first one is transcription enzymes. So, what are the transcription enzyme? We have the ribonucleic acid polymerases or RNA polymerases. So first one is RNA polymerase 1. RNA polymerase 1, it makes, what's its function? It makes RNA the most common or rampant excessive or RNA, which is known as the ribosomal RNA, and it is present in the nucleolus. Then we have we skip RNA polymerase 2, we discuss RNA polymerase 3, which also makes ribosomal RNA. It's part 5 S part, and also transfer RNA, which we'll discuss in the transfer in the protein translation. Now RNA polymerase 2, it makes first most important messenger RNA. It makes micro RNA, which is known as mRNA, which is important in splicing process, and micro RNA, and then it form the small nuclear RNA or known as SNRNA, or and it is the main RNA which is used in splicing process. Micro RNA are used in processing of messenger RNA. So messenger RNA, snRNA, mRNA, transfer RNA, rRNA. So RNA polymerase is also open DNA strand, open the DNA primer site, promoter site for RNA polymerases to bind with. So it is the opener. Also it produces three different type of three different type of ribonucleic acid. So we have discussed 1 2 3 different enzymes. Now there is a death cap mushroom, which releases alpha amanitin, and it actually inhibited RNA polymerase 2. If RNA polymerase 2 is inhibited, then there is no messenger RNA, no splicing, no processing. So there is protein translation is inhibited. So the person is suffering from acute dysentery, and there is a severe type of hepatotoxicity. Just MCQs, approximately 1 or 2 MCQ are related from this messenger RNA topic. Also in these messenger RNAs, rifamycin, their rifampin and rifabutin, rifampin and butin its type, it inhibit prokaryotic RNA polymerase, and which RNA polymerase? Which is DNA dependent RNA polymerases. And in prokaryotes there is only one type of RNA and having multiple segments, so which perform different type of function of RNA polymerases. Then we have actinomycin antibiotic, actinomycin or dactinomycin. It inhibits both prokaryotic and eukaryotic RNA polymerases. RNA polymerases, they do not need, so these are the medications. So RNA polymerases do not need proofreading. and they can initiate any chain processing. Can initiate the chain processing, but they do not need any kind of proofreading. After discussing different enzymes, we have now in the genetic code, let's suppose this is the code. First we have an enhancer or silencer part. In the end we have also a silencer part. After silencer part we have a promoter region, and the promoter regions are the cat and the Tata box. Promoter, enhancer and silencer. We have in the end we have the enhancer or the silencer part, promoter part, then we have the silencer part. And what promoter part do? The cat, it is upstream at the 75 base pair upstream from the initial exon. And we have promoter and the Tata box, which is 25 base pair upstream, so it is upstream from the main gene, coding gene. Enhancer, so promoter is the locus, these all are the loci on the gene. So the locus which is on the DNA, it's the region at which RNA polymerase 2 and different transcription factors they bind and do their functioning. So it's a binding site for RNA polymerase 2. We have discussed here that it opens the promoter site. So RNA polymerase 2 bind and different type of transcription factor binds at the promoter region, cat and Tata box, and it is upstream. Whereas the loci which are enhancer and the silencer, they are the binding site for what? For the regulatory proteins. So silencer and enhancers are actually the sites where the regulatory proteins bind. Which regulatory proteins? The activator regulatory protein and the repressor regulatory protein. At silencer, the regulatory protein is repressor. At enhancer, the regulatory protein is activator. They both bind. They are present in activator protein. It actually increase the speed of transcription, and silencer, it actually decrease the speed of transcription speed or transcription. And the presence of enhancer and the silencer, they are present downstream, they are present upstream, or they are present within the same intron. So they are present either upstream, they are present either downstream, and they are present either within the intron. So the two MCQs over here is the upstream, the positioning of these, the processing, what enhancer do, what silencer do, and what is cat and Tata box loci. So these are the three MCQ in this 4 to 5 lines. So this is a actual eukaryotic gene or eukaryotic gene which is going to make a protein. So we have known that there is exon part and there is an intron part, and the exon part starting point and intron part, they have the, and exon has the start codon UAG and intron has the stop codon UGA. After this we have a polyadenyl tail. This is the point where transcription starts, and this is the point where actual coding region of the protein is started. Then we have the intron, then we have another exon, and after that the stop signal come, and the polyadenyl chain formed at the end of the intron, and then at the end of the transcript, and then we have a release factor from silencer loci, and the replication happen. So this is transcription happens. So this is the important enzymes. These are important loci, and this is the main typical intron or exon transcript of the messenger RNA. Then next we have this transcription. So now we have initial transcription transcript, which is actually an silencer region. We have a poly A tail region. We have a stop codon UGA. We have exon 2 intron, then again exon, the Tata box or the cat region, and then we have the enhancer or silencer region. So this is actually a typical example of a gene in which there is which is recently replicate from the DNA, and now it is transcribed into initial transcript. So the initial transcript is the heteronuclear, it is still in the nucleus, heteronuclear messenger RNA, which is the initial transcript. So in the nucleus there is a nuclear processing, in which we add this heteronuclear RNA initial transcript, we cap it, we tail it, and we splice it, and then we make it a mature RNA, messenger RNA, and exit into the cytoplasm. So the nuclear processing happen in the nucleus at the adding that cap at the fifth end of this messenger RNA. Like this is the fifth end, this is the 3 end. So adding cap, which cap? We add 7-methyl guanosine cap at the fifth end of the messenger RNA. We add a poly A tail or polyadenylation process at the 3 end to save it from cytoplasmic enzymes which are at the brink of eating that messenger RNA, which is newly transcribed from the gene. So in order to save that particular transcript from that savage proteins, we have to make it cap and tailed. So in order to we package the main code into cap and tail and splice into this part, so that it is safe from that proteins. So it is safely goes into the cytoplasm and then processed to produce proteins. So capping and tailing, and also we do splicing, alternative splicing, which we'll discuss in the last, by the SNRP protein to make the mature messenger RNA, and then it exit into the cytoplasm. Over there, the cytoplasmic have a cytoplasmic processing bodies or P bodies, which actually control the quality control of messenger RNA. And these bodies, they have exonucleases, they have decapping enzymes, and also they have microRNAs, which only microRNA it either degrade the protein or save it or store it for future translation. So we have the exonucleases enzyme over there, and we have known that exonucleases check all the errors. We have the decapping enzyme which remove this guanosine from the initial transcript messenger RNA in order to sequence it, in order to translate it into protein. And we have also a microRNA which helps either in the end degrade that messenger RNA or store it into the P bodies in order for future translations. Then we have discussed the enhancer silencer part, we have discussed the promoter parts, we have discussed the poly A processing. Now we discuss the introns and exons. So exons and introns. Exons are the actual genetic information of a particular protein, and which encodes or codes or expressed or translated it exit the nucleus, whereas the introns are just the intervening parts or the sequence. They do not encode or do not express, and they remain into the nucleus. So why we have remain in the nucleus? Why we have these introns? Because these introns are important in the regulatory gene regulation process, gene regulatory expression process. Over here we have now we make genetic information into heteronuclear RNA, which processed into the nucleus to produce messenger RNA, which is further processed into cytoplasm to remove the exons and introns and make different types of proteins. Particularly the cancer evade immunity, host immunity. How? By using alternative splicing process, alternating splicing process in order to splice out the exons for the FAS receptor protein in order to evade from that FAS receptor ligand apoptosis process or programmed cell death process. So this is the mechanism of cancer cells which they use to evade the body immune response and causes different type of cancers. So now how this splicing process actually work, how alternative splicing happen. Alternative splicing happen by first we have a 5' and then we have 3'. At the 5' we have a particular type of exon 1. At 3' we have also exon 1, and in between these two exons we have intron part, and we need to remove this intron part in order to combine these both exons. So we have a protein known as the small nuclear small nuclear RNA or small nuclear ribonucleic proteins which came and bind to the 5 end of the intron part, this part, and we have also a U2 SNRP protein which come and bind to the branch point. First protein bind to the 5 end of the intron at the this codon, and the next is bind to this branch point, phosphate, and this. So this is the intermediate exon intron part, and we have to cleave this. So this branch point is actually be the main central point over which this whole cleavage happens. So when the cleavage happen at the 5th end, the larate or this loop is generated. Larate or this loop generated. And when the cleavage happen, SNRP cut this, and over here the phosphate bond cut this. So this hydroxyl is bound to the other part of the exon. So now exon phosphate to the other exon, and the larate part is actually the this branch point over the U2 ribonucleoprotein is present, and they make a bond with the phosphate of this UG intron, and the remaining section is over there, which is EG hydroxyl group. So this is actually the loop which is present over here, which is known as the larate, and this loop is important in order to splice out this protein. So when the SNRP or SNR and RNA it bind to the 5th end of the, it binds to the intron, then we name it as a spliceosome. So this is how the introns and exons are spliced out in order to make a particular type of messenger RNA code, which then produce different types of protein. So exon 1 and exon 2 are binded together. So now protein, this protein and this protein is formed. It may be anything. If there is an anti-U1 small nucleoprotein is present, antibodies against this SNRP protein happen, then it can cause rheumatological conditions, for example SLE or mixed connective tissue disorder. And there is also a condition in which there is alter in the SnRNP assembly. If SNRP alter, change in their assembly, then it can leads to decrease in the synthesis of survival motor neuron protein, which can cause congenital degeneration of anterior horn spinal cord neurons, causes the floppy baby syndrome, whose symptoms are hypotonia and symmetrical weakness. So this is these two MCQs are important. Which antibody causes SLE? U1 SNRP, and SMN protein assembly alteration can cause the floppy spinal muscular atrophy, it can cause this. So this is all about the RNA processing and RNA polymerases. We have discussed RNA polymerases. We have discussed the loci, promoter, enhancer sequences. We have discussed nuclear processing of initial transcript. We have discussed the cytoplasmic processing. We have discussed the exons and introns, and we have discussed all the splicing process.